So I think one of the ones that I think is very much sort of front and center is getting to an off-the-shelf version of a T-cell therapy that has the same potency and potential durability that we're all after in solid tumors. We've actually been working on that ourselves for some time. 4 years ago, we had signed a small technology collaboration with a company called Universal Cells, which had an AAV gene editing platform for editing stem cells and removing sort of certain molecules that would make the cells be rejected. So we took that platform, worked with them, and then were able to develop a process, a proprietary in-house process for differentiating stem cells into T cells. The reason we took that view, there's a different way of producing off the shelf where you take a donor source of mature T cells and edit those. And the concern or the view there is those cells then disappear. They get depleted by the body over time, whereas a stem cell that's been differentiated into a T cell can survive and can persist in the patients in the same way we hope as the autologous products do today. But the idea is that you have your edited version of the autologous product effectively sitting on the shelf, so that when a patient is diagnosed, that product and they're tested for their targets. What is it they express? Have we got a TCR construct in an off-the-shelf version? It basically can be shipped, thawed, and administered. And I think that first of all, that changes the whole dynamic in terms of access and availability. It changes the dynamic in terms of costs of good versus costs of developing the therapy and pricing. So I think that is potentially hugely disruptive. There's still quite a bit of the biology to determine, and we've done a huge amount there to produce functional T cells from edited stem cells differentiated to T cells, which is a massive step forward. Now it's about scaling that and bringing it into the clinic and doing that on with multiple different products. So I think off the shelf is going to be very disruptive if it's as effective and impactful as the autologous products are beginning to show. So that's one area. The other, I think, is going to be screening diagnostics and having high resolution understanding of a patient-specific tumor and targets and potential other molecules that will impact if you like, the nature of their particular tumor and the second generation modifications that could be important. And again, hopefully that, the right version of the T cell will be, potentially other cell types will be available off the shelf and administered in a more real-time way. And obviously then linking in with other modalities, checkpoints, etc., which obviously also impacting generating a sustainable immune response to control cancer as opposed to responses and progression, which is somewhat where we are at this point. But even though that is a massive step forward.